Today, we're joined by Monique McHenry, and she's an assistant professor of pharmacology here at the University of Vermont. And we had heard from a number of you that have been involved with our webinar series that you wanted to know more about the chemistry of cannabis and the research and science that is behind how it works in the human body. And so we reached out to Monique and she agreed to come and present the latest and greatest research to all of you so that you have more information in your hemp growing ventures. So welcome, Monique. Thank you for joining us today.
Thanks for having me.
Yeah.
So welcome everyone to this webinar on cannabis plant chemicals. The next slide is going to talk about my disclaimers and disclosures. So if you can fast, yeah, there you go. So what we like to talk about all the time when we're discussing investigational new drugs not approved for use in the United States is that although I am faculty at UVM, the materials presented here represent my own findings, views, and opinions, and they shouldn't be taken as a statement, position, opinion, or endorsement by the University of Vermont. And so the next slide is going to talk about basically what I was hoping to cover today. And it's a big list, even though it only has 4 objectives. And so I hope we can get through it all. Again, please interrupt, or you can save your questions to the end too. So we're just going to briefly cover why plants produce chemicals. We're going to review facts and hypotheses on the production of phytocannabinoids, especially discussing the variables that affect the amount that we find in cannabis-based products. And then we'll talk about how these chemicals affect the human body. And so the next slide, thanks. We go into plant chemicals. So there's 2 categories of plant chemicals. You have primary metabolites. These are your sugars and fats. These are synthesized for essential functions. And then you have the secondary metabolites. These are like cool chemicals that are synthesized for specific functions. The 5 most common in plants are alkaloids, which we'll discuss briefly today, terpenes, glycosides, phenolphthalides, and proteinaceous compounds, which we're not going to talk about at all. And then on the next slide, I'll give you a couple examples before we move into cannabis, right? So an example of an alkaloid is morphine, right? You can see that wounded fruit smack center in the middle of the slide. And that is what's going to contain your morphine, right? It's found in the opium poppy. There's actually 24 different alkaloids in poppy. And the whole thought, from a plant perspective is why was this produced? Obviously not for pain relief in humans, but probably as a herbivore deterrent, because it doesn't taste very good. And as you know, some of the synthetic derivatives like opioids are great pain relievers in some applications, but they can also lead to addiction. And then the next slide, we talk about an example of a terpenoid. And a terpenoid not in cannabis is lycopene. And lycopene is found in the tomato. It's a tetraterpenoid. So that means it has 8 isoprene units. For those of you interested in chemistry, right below that giant red tomato, you'll see isoprene units stacked together, forming lycopene. It gives tomato their red color, and it's hypothesized to be an attractant for animals. So instead of deterring them, it would attract them to the ripened fruit instead of the immature green fruit. And then that would aid in seed dispersal, right? So they poop out seeds and you move them further away from the plant. In humans, it's hypothesized to reduce risk of cancers and lung disease. So now if we quickly move into, on the next slide, just talking about why plants, why would they need to defend themselves? Well, they can't bite or make a fist, right? They're sessile, so they're fixed in one location, and they can't free predators. So they need to have another way to defend themselves. One method is that they have collectively synthesized this plethora of secondary metabolites to defend themselves against herbivores and microorganisms, and to adapt to different types of abiotic environmental stresses. And if we go to the next slide, what we'll see is some ways in which plants defend themselves with or without chemicals. And we're going to focus right in on trichomes, and I'll tell you why in a sec. Trichomes are plant hair, and they are active as defense mechanisms. Catherine, if you hit the advanced button, you should see, yep, an image come up that says hard trichomes. That's an example of hard trichomes, and they can cause a physical injury, right? If you touch that, it's going to hurt. And then again, if you advance one more time, you'll see a picture of sticky trichomes. And those can trap insects, but they can also store chemicals. And those are the ones we're going to talk about today. And then you also have fragile trichomes. And if you hit the advanced button, you should see a nettle pop up. And those dispense chemicals, and they actually kind of act like a hypodermic needle where the chemicals are stored and then they're inserted right into the unlucky person. And so today, we are going to focus on the sticky ones, or the glandular trichome. And this slide is full of a lot of information we're not really going to talk about. I'm just going to mention it, and it's going to be recorded for your review if you want to really delve into the plant morphology of, glandular trichomes. But they're just multicellular structures, so they have more than one cell, and they consist of differential areas, stalk and the base. And the one important thing to know is they are not unique to cannabis. They're present in about 30% of plants, and they commonly function to produce, store, and secrete secondary metabolites, right? Some common uses of these metabolites are natural pesticides, food additives, and pharmaceuticals. Now, one thing to note is in cannabis, the phytocannabinoids we're talking about are found here in the glandular trichome, and they are unique to cannabis. So although about a third of all plants have glandular trichomes, only cannabis has glandular trichomes with these interesting chemicals, the phytocannabinoids. So if we move on to the next slide, I'm going to quickly review all the cannabis chemicals. So that top picture you see, again, is the glandular trichomes. And the graph you see to the right is all of the chemicals in cannabis. If you look at the whole leaf, right, not just the glandular trichome, you'll see a bunch of different terpenes, and then highlighted in orange there is your cannabinoids. You also have flavonoids, pigments, sugars, fats, chlorophyll, waxes, and the list goes on. Now, if you extract cannabis into a raw form, and what you see in your bottom picture, you advance it one more, we'll get another graph on there. So Catherine just hit the advance button one more. Yeah, there we go. And you'll have your hash oil or your essential oils. And those still have sometimes different terpenes in them, and flavonoids and pigments and chlorophylls. That's why you're seeing color to it, right? They can also have fats, waxes, and cellulose. And then Catherine, if you advance it one more time, what we'll see is if you zero in on the cannabinoids, they're kind of devoid of color, right? Because you have white here. And so on the next slide, I just want to mention one thing is sometimes people casually talk about cannabinoids, right? Today, we're going to focus on the bottom image, the phytocannabinoids. Phytocannabinoids are found in the cannabis plant. There's two other types of cannabinoids, right? Our body, the human body, produces endocannabinoids. And then there's also synthetic cannabinoids that are produced in laboratories. And an example is sesamet or nabeline, which we'll discuss a little bit about. There's lots of synthetic cannabinoids that are used on the black market, and a few that are approved for use in the US. So thank you for advancing it. One of the questions as a botanist for me was, why did cannabis evolve to produce these phytocannabinoids? And this information helps us better understand also how to produce consistent cannabinoids in a cannabis crop. And so I'll go through it relatively fast. Again, these slides all have evidence-based data on them. So they're all, publications, sometimes from a long time ago, like this one from 1987, where Linden and colleagues found that cannabis evolved potentially to produce phytocannabinoids as an environmental response, right? So what this graph is showing you, that phytocannabinoids increase when you increase the levels of UV radiation, or specifically THC increases. So they hypothesized that phytocannabinoids therefore acted as a UV protection. And then on our next slide, what we'll find is more evidence that phytocannabinoid evolution was an environmental response. And there's two studies. One was recently done by a graduate student in 2018, Kaplan, and another is older, somewhat anecdotal data, but it is evidence-based data, so we're including it. And what we find in these two studies is that major phytocannabinoids increased when you apply an intentional drought. And so basically here, 7 weeks into the reproductive stage, what you'll see is the red arrow points to the significant difference found between plants exposed to drought and a control in both THCA and CBDA, so the major phytocannabinoid levels. And in the lower experiment from Sharma in 1975, they reported a greater glandular trichome density on leaves of cannabis that grow in hot, dry environments. And you can see that in the bottom side, right? Highlighted in red for hot, dry climates and blue for cool, humid climates. And so if we move to the next slide, we'll see another potential reason why cannabis evolved to produce cannabinoids, potentially as a stress response. So again, an older study where you see correlation between low plant height and high phytocannabinoid content. And Catherine, if you advance one more time, I think we should get some arrows and boxes. There we go, maybe one more, we'll get one more arrow, hopefully, there we go, yeah. So on this slide, you can see data that supports the plants grown in different soils produce different amounts of your phytocannabinoids, delta-9 THC and CBD. And I've highlighted magnesium because it seems like the lower magnesium you have, the higher THC and CBD. And I think these are all theories that have gone through the cannabis growing community. And if you look in older literature and newer literature, they actually do have evidence to support that this is true. And if we move on to the next slide, this is more of a far-fetched theory of why cannabis evolved to produce phytocannabinoids, but there is a study out there that says they are produced as an insecticide. They compared THC-rich cannabis and found that there was some fatalities observed in a tiger moth from THC-rich Mexican versus CBD-rich. So to advance to the next slide, we'll see another possible sound hypothesis in the evolution of phytocannabinoids is that they evolved as a competitive advantage or a chemical plant defense against other plants. So here on this slide, I have two reports that support the hypothesis that cannabis participated in chemical warfare, right, against other plants, as cannabis extracts inhibited the germination of lettuce and decreased the germination of monocots. And I think we have one more possible hypothesis for why cannabis evolved to produce phytocannabinoids, and that is as a disease prevention. So we see evidence that phytocannabinoids could harbor antibiotic properties, and that antibiotic properties assist in the overwintering of seed. So although you don't see phytocannabinoids produced on the seed, you definitely find them on the seed when you're measuring cannabinoid levels because they fall off the inflorescences. And so we advance the slide. I'll just give a quick summary of what we talked about, right? So cannabis evolved to produce phytocannabinoids as potentially an insecticide, a competitive advantage, a disease prevention, an environmental response, or a stress response. And then our next slide, we go into another question that I think is really important to talk about is why are the amounts of phytocannabinoids inconsistent? And we find a few different factors that can correspond and some places correlate to those inconsistencies. And the first is that plant organ matters. And so Catherine, if you advance a little bit more, we'll get some red boxes that get highlighted. So, oh, sorry. Yeah, one too many, there we go. So what you see on this is the highest amount of THC and CBD is found in the flower, right? The red box to your right. And what you're seeing is the numbers and the top box, THC and CBD in the drug type are going to be highest in the flower when you compare it to the leaves or the stem. And then of course, pollen is very low and seed is very low. And it's probably just a dusting because they don't generally have glandular trichomes on them, as well as the root. So if we advance one more time, we should get to the next slide. There we go. And what we see is the amounts are also inconsistent because the age of the plant determines how much THC and CBD and CBG you will find in the flowers. So on this slide, we've included numerous bar graphs from 2 different studies that report phytocannabinoid increase as the plant ages. So the top 2 graphs are THC concentrations, and they're represented by the bar for different times in the life cycle of cannabis, increasing until you reach a plateau. And then the bottom 2 graphs one represents CBD and one represents CBG. And what you see with CBD is it continues to increase throughout the life cycle until they stop their measuring point, where CBD reaches a peak concentration and then decreases for the remaining measured days. And one kind of quirky study, but it's interesting to note too, is that as the size of the size of the glandular trichome head increases, so does the amount of THC, which seems pretty apparent, but it is good to have a study with evidence-based data published that show that this is true. So the smaller your gland, the smaller the amount of THC you'll find. And then we find that cannabis grown with different fertilization concentrations also produces different phytocannabinoid ratios. And this again was done by a graduate student, and he experimented with the fertilization rate and he reports increased rates of fertilizer application, increased yields of THCA, but that the yield continued to increase with additional amounts of nitrogen, although the phytocannabinoid concentration didn't necessarily continue to increase. So this could suggest a dilution effect, right? Where the plant has more mass, but you don't get more phytocannabinoid production. I think recently, and maybe even UVM extension, Heather's research has found this isn't necessarily always the case, that you can get a higher yield and correlate with a higher concentration of phytocannabinoids, as you increase the fertilizer rate. And they did find what they call the sweet spot, where you have a substrate rate of about 212 to 261 milligrams produced the maximum amount of THC and CBG. Okay, so the next slide, thank you, Catherine, you have it up already, is why are the amounts of phytocannabinoids inconsistent? Well, position on the plant matters. I think a lot of people know this that are growing cannabis, but the uppermost part of the plant, the apex, so the highest inflorescence, produces more phytocannabinoids. Here we go. We have a publication that supports that. And you can see those bar graphs, decrease significantly when you go from the uppermost branch to the lowermost branch. And something that isn't always studied is light quality. So a study that came out from Italy in 2018 showed that the phytocannabinoid yield changes with different light sources, right? So the top right graph represents the wavelength of the different lights the authors used. And so if you note the blue arrow, which if Catherine advances should show up, identifies the light source NS1. And that light source has a wavelength that has the highest amounts of blue and UV light in the experiment. And if you follow the blue arrows, if Catherine advances one more time, we should see them show up. Hit it 3 times, please, down on the lower graphs. And there you can see that NS1 has the least amount of gram per flower, but the highest percentage of THC and CBD. So therefore, the bottom right graph, you can see that it has the highest amount of cannabinoids per gram of flower. And older research also reports from 1980s that the levels of UVB radiation correspond with increased levels of THC. So it seems like the more blue spectrum you have, and if you add UVB, you see higher amounts of these major phytocannabinoids, THC and CBD. And the following slide, we start to talk about terpenes a little bit, because I know people are not just interested in these major phytocannabinoids, THC and CBD anymore. And this study, again, in 2018, found that not only does light quality matter, right? So they're finding a lot of difference in their, the different lights they're using, but the illuminance matters. So basically they supplemented the lower branches, so the second canopy in this study, of the inflorescence with a green-blue light. And the different wavelengths from the light treatments are represented on that bottom left graph. And they report that the additional green light increased the terpene concentration, as you see on the graph, and the blue arrow is pointing to it, in 3 different treatments. So RGB is the bottommost row on the graph, identified with that blue arrow, right? So if we go into just a couple of slides, if you advance, from UVM extension research with Dr. Darby's team, what you find is you also have to remember that phytocannabinoids are not consistent across different cultivars. And so if you advance one more time, I think we'll get something on that slide. There we go. Thank you. That says, the parameters you can control in this study were what was harvested, the amount of light, the type of light, the soil composition, the available moisture, and what age they were harvested in. What you didn't control for was the genetic composition, right? So it's just different cultivars. And of course, they're going to have different levels of THC and CBD, which you see in blue and green. Oh, well, green is the ratio of CBD to THC, and blue is the total potential CBD on this graph. And so if we advance one more time, it's another slide from UVM extension research, is that growing environments. So you're controlling for the organ harvested, the genetic composition, the soil composition, the available moisture, the harvest age, but what you're not controlling for, so the different variable, is light and temperature. And I think this is called outdoor and indoor for CBD, and you're seeing the red is the indoor. It's just a caterpillar that I believe Heather's team put up. But you see, sure, there's inconsistencies. You have the exact same cultivar here, and what you're seeing is that indoor, you're going to see higher amounts of CBD than quote unquote outdoor. And so the next slide again brings in the terpenes. And so there's very little terpene data, but this is a good study, because what it shows us is, as we're learning more and more about the things that affect phytocannabinoids, we can kind of assume that they'll be affecting terpenoids as well, because the levels, so the total amount of phytocannabinoids and terpenoids, have shown to correspond. And I know certain terpenoids, will differ depending on the day, but in general, you can say they correspond to the phytocannabinoids. So the research we just talked about that affects phytocannabinoid consistency should also be thought of to affect terpenoid consistency. And the next slide is the answer to a question that a lot of people ask. So is it possible to grow cannabis with reproducible terpenoid levels? And a study that came out about 10 years ago now says, yep, you can, but you have to make sure your genetics are standardized and your environmental conditions are standardized. And what this graph is showing, and if you advance a few times, maybe just once and see, yep, perfect. Okay, go ahead. Yeah, there we go. Thanks. You'll see that the 11 strains that they grew are all represented by different numbers. And so what I've done is colored kind of the cluster of numbers with different colors and highlighted them in red. And they tested for 36 compounds and just did this fancy multivariate statistical analysis to prove to us that, if you look down at the bottom, you see all those threes clustered together, meaning they have more in common with each other than they do those other numbers. And so you can say, okay, their chemical composition is there's a discontinuity discontinuity between all the 3s and all the 4s, for example. So the 3s were able to be consistently produced with the same chemicals that distinguishes them from strain or cultivar 4. Hopefully that makes sense. Okay. So there's just a few more slides here discussing why the amounts of phytocannabinoids and terpenes are inconsistent. And these slides are after harvest. And again, this is the UVM extension research slide in a project that Heather's team did a couple years ago, where we had them measure the total potential CBD in an ambient and 80 degree and 105 degree. And I think there's some caveats with this drying, but basically post-drying alters cannabinoids and definitely alters terpenes, right? And so demonstrated in this graph, you can see the red and blue bar represent the total potential CBD with just the 2 variables I mentioned, basically you find the highest amount of total potential CBD was measured from the flowers that weren't dried with a dryer. And there was a significant difference. But the flowers dried at the warmest temperature had the lowest amount of CBD. And they also did research with keeping them on the stem and not on the stem. So flowers only is blue and flowers on the stem. And that is red, and that seemed to increase the amount of CBD if you leave it on the stem. So this next slide shows that UVM research does mirror what is found in the literature, and that post-harvest storage matters. And this is a combination of work, both from 2009, and it shows that post-harvest storage alters phytocannabinoids and terpenes. So these studies report that air contact caused the decreased levels in THC in both light and dark conditions. So if you want to go back and look through these graphs and read about the different conditions, what you'll notice is if you're looking at THC, you'll see a slight decrease over storage months, which is your x-axis, in the conditions where you have light and air exposure. Numerically, they're suggesting a continual decrease of up to about 74% of the original concentration after 4 years. What I didn't show on this graph is their CBD data. And I know that's probably more interesting for this group. And the CBD data, the concentrations weren't really as affected, right, when you have different storage conditions, but they are prone to oxidative degradation, which means exposure to air. It does decrease or degrade the product. So I think we just have 2 more slides on post-harvest storage. And this one again is from UVM. And this, what we see is post-harvest storage alters phytocannabinoids and terpenes. That the red and blue bars here represent the amount of total potential CBD with 3 variables: temperature, exposure to air. We do see a small decrease in CBD in most treatments over time. We don't really see that one treatment outperforms the other treatment. And that corresponds to the previous slide where we just talked about there isn't much difference in CBD, except there's potential oxidative degradation going on. But besides that, storage doesn't seem to alter as drastically the concentrations of CBD as it does THC. And then we see that most terpenes, now we're talking about terpenes, exposed to air suffered from evaporative losses, which is as expected, right? Because a lot of them are volatile chemicals. And I would encourage anyone interested in this to use this publication and look more into it. Don't want to bore you and bog you down with the science of it. But, basically terpene decrease or increase over time without exposure to air. And they're basically undergoing more complex chemical reactions. And that's what you might be seeing in these graphs. But it's important to note that people are looking at terpenes too, and how post-harvest storage affects the terpene levels. And so then the next slide is just a quick summary of what we talked about, right? So the factors we discussed as potential drivers in phytocannabinoid inconsistency with evidence-based data supporting it are changes in phytocannabinoid production are due to the organ harvested, the harvest age, the position of the flower on the plant, the soil composition, the light, both quality and illuminance, the genetic composition, and the drying and storage. So, basically, why does this matter, right? Why do we care about all these factors? And it's because they affect label accuracy. Research done in Vermont a few years ago found label inaccuracy. So, sampled them. That was basically what was in the stores in 2017, and 10 Vermont brand products. And we found that the national brand labels were far more accurate than the Vermont brand labels. Of the 14 national brand products, 11 were accurately labeled and 3 were over-labeled with 1 under-labeled. And of the 10 Vermont products, 1 labeled and 3 had no labels. So if you advance a little bit, Catherine, please, we'll see some bullet points pop up. The best one is all products contain CBD. Only 1 product had levels above 0.3% THC, and it was only at 1% THC. It was a cartridge, and I don't think that was malicious. And all in all, Vermont products plus national products, you see 50% were labeled accurately, but then the last bullet point does tell us that only 17% of Vermont products were labeled accurately. And then we look at the delivery method, and that's what you see color-coded on this graph. It affects the accuracy of the labeled dose. Capsules were the most accurate, followed by tinctures, but the concentrated and cartridges were difficult to compare, basically due to limited labeling, like the dose wasn't necessarily labeled on there. Tinctures that recommended a dose of a full dropper or a single drop, 1 full dropper versus a single drop were more accurate than tinctures that recommended like a multiple drop dosing, because it's really hard to be consistent with what comes out of your dropper. So the whole dropper is more accurate for a single drop than like 10 drops. And so if we advance, what we see is that, advance the slide, we see that the label inaccuracy we found near literature. So a study by Bob Miller and colleagues from products purchased online in 3 different forms, right? So you have oils, tinctures, and vape liquids, found that 31% of products were labeled accurately, and 43 were underlabeled with 26 overlabeled. Vaporization liquid was most frequently mislabeled, and oil was most frequently labeled accurately. And that's what these numbers are showing. And so, Catherine, if you advance twice, I think we'll get through our bullet points and on to the next slide. Thank you. Or advance, yeah, there we go. And why do we care about this, right? Why do we care about label accuracy, phytocannabinoid inconsistency due to post-storage and growing? And the reason why is because phytocannabinoids impact humans. So if you have a different dosage and your label is incorrect, you're going to have a different effect on humans. So if you're creating your product for basically not just humans, but any mammal, you want to make sure that you have consistent production and accurate labeling. So, Heather asked me to come here to talk about how phytocannabinoids do affect the human body. And I have to say a disclaimer here, I am a plant biologist, but I do work for the Department of Pharmacology. So this material is something we now teach. But my pharmacology, it's in plant biology, but I do understand this material, so I will willingly talk to you about it. And so basically, cannabis impacts the human body through the endocannabinoid system. So phytocannabinoids have a pronounced neurological effect on receptor specifically, perhaps as old as the least common ancestor of all bilaterans, right? So everything that has a right and a left identical side. So animals with bilateral symmetry, and that would be over 500 million years ago. Cannabis diverged from hops probably about, anywhere reports are like 12 to in the 20 million years ago. So the evolution of a endocannabinoid system with receptors way predates the production of phytocannabinoids. But this led to an important research question: is how do humans respond to the chemicals produced by the cannabis plant, millions of years after we produce those receptors? And that line of research led to the discovery of an entire biological system. In humans, we call it the endocannabinoid system. Turns out we produce our own endogenous or naturally occurring cannabinoids called endocannabinoids. These act on cannabinoid receptors that are found in abundance throughout our body, including neurons and immune cells. And then recently, we've begun to understand the fundamental basis of the system and the consequences of its activity. For example, like runner's high is something that's hypothesized to be responsible. The endocannabinoid system is responsible for runner's high. So the endocannabinoid system consists of cannabinoid receptors, endogenous agonists, and agonist metabolizing enzymes. And so if we advance the slide, we're going to go into a little bit more detail about the classical cannabinoid receptors, and they are CB1 and CB2. And CB1 is primarily expressed in nervous tissue and specifically in neurons, right? That receptor, CB1, is also found in adipose tissue, blood vessels, gut, testes, uterus, and elsewhere. But thinking about the major phytocannabinoids and their relationships to these receptors, so THC is a partial agonist. So what does that mean? It means it accepts the key, right? If you think of a receptor as a lock, THC, the key can fit into the lock of CB1, while CBD does not fit into the lock. In fact, it blocks the lock. So I guess it fits, but it blocks it, right? So so that's the difference, right? CBD partially fits in the lock. THC partially fits in the lock. CBD blocks the lock of the CB1 receptor. Now, CB2 is the other classical receptor we're talking about, and it's primarily expressed in the immune system. So if we go back to talking about CB1, and what this image shows on the slide is an audio radiograph of a rat brain slice. And it's exposed to a titrated synthetic cannabinoid. If you're interested, it's called H3CP55940. So we're talking about a synthetic cannabinoid here. And what you see is that the CB1 receptors are distributed unevenly in the brain, and that this distribution correlates with the behavioral effects of THC. So in this image, the highest densities of receptors highlighted, right, that are acting on this synthetic cannabinoid are highlighted yellow and orange. So those bright colors, yellow and orange, and even kind of fluorescence. Campus, which affects the short-term memory. But you see really low densities. So you see it's black or kind of red in the brainstem. And the which is basically the cardiorespiratory center. So this probably accounts for the lack of lethal effects from cannabis overdoses, because you don't you don't see the receptors activated in the brainstem. There's no receptors there. And this is very much unlike what you would see in an opioid receptor distribution. So even though this is a rat, right, so it's a preclinical study, it can give us an indication of what's going on in the human brain too, when it's exposed to a synthetic phytocannabinoid. So on the next slide, basically, I just want to talk a teeny bit about the basic pharmacology of CBD and THC. So what happens when these major phytocannabinoids enter the human body? So the first step is absorption into the blood. And there are a few different factors that you might not think about that influence that absorption of the drug, THC or CBD. And that's the mode of absorption. So are you inhaling it, or are you swallowing it, or is it going directly into your vein intravenously? Also, what you ate, did you just eat something very fatty before you administered the drug? If you're sick, the use experience, right? So do you have a tolerance to this particular drug, THC or CBD? And then the chemical properties of the drug, and are there any absorption enhancers? Is there, a nanoparticle of whey, for example, that might increase the bioavailability? So in a report from 2013, where these researchers are using an oral or oromucosal, so they're either swallowing it or they're dissolving it under the tongue, which actually have a very similar bioavailability, and we'll talk about that later. You find from the graph, THC on the top, CBD on the bottom, where time is the x-axis and concentration of the drug is the y-axis. They report that CBD is generally kind of less absorbed, right? For example, CBD has a smaller maximum concentration in the body, as you can see by that smaller peak, and that's the Cmax. But CBD takes slightly less time to reach that concentration, and that's the Tmax. So you have a shorter Tmax. And then CBD takes longer to leave the blood. And that is represented by a larger T-half. So basically like the half-life of the time it takes to half of it to leave your blood. And if we go to the next slide and we talk about bioavailability, so basically when we're talking about absorption, so a quantification of how much is absorbed, that's bioavailability, or the amount that gets absorbed. The quantity and the duration of THC entering the blood and CBD entering the blood are dependent on the administration method. So these numbers you see at the top are THC. And what we see is that oral and sublingual have shown similar absorption profiles, right? And sublingual, to note, is under the tongue and it's getting absorbed by those tissues in your mouth. And oral seem to be the same, right? But if you're putting something directly into your vein, 100% of it is going to be absorbed. If you're inhaling something, 10 to 30% of it is going to be absorbed. And then oral and sublingual have the least amount absorbed, right? 6 to 10%. And so I've put, and we're talking about delta 9 THC in this study. And I've put down a graph for two reasons. One, because we didn't talk about transdermal because that data was not in the study. And we haven't really talked about delta 8 THC, which is now becoming a popular minor phytocannabinoid. And what you see is that there's two important notes. One, there's little published research about minor phytocannabinoids as well as transdermal. That's up and coming, right? So the minor phytocannabinoids like delta 8 and transdermal application, we're just learning more about now. But then the second is that we see, yes, this isn't a rat, but there is transdermal absorption of delta 8 THC. And the curve like very different from the curve we looked at in the previous slide is it takes a long time for the absorption to go into effect, and it's kind of a flat curve, right? You don't see this really peaky part of maximum concentration in the blood. So what does that mean? Well, we're going to have to find out. We'll have to do more research on both transdermal and delta 8 THC, along with other phytocannabinoids. But if we move on to the next slide, we can focus more on CBD. And again, this slide is preclinical, meaning it's in non-humans. And the research is reporting that cannabidiol, so CBD, activates some non-endocannabinoid receptors, which is totally normal. Many plants, activate receptors in the human body, which have a completely different function than being activated by that plant compound. And CBD activating the TRPV1 receptors is one example. But TRPV1 is a receptor that's found in nervous tissue. It's involved in the feeling of pain. And this receptor's activity can be decreased with overactivity, meaning it can be desensitized. And that will decrease the perception of pain, right? Leading to this analgesic effect. So CBT acts by delaying the reuptake sometimes of endogenous neurotransmitters, such as anandamide and adenosine, and by enhancing or inhibiting them to other protein receptors. But I just wanted to note that the phytocannabinoids don't only act on these classical CB1 and CB2 receptors that are found and considered part of our endocannabinoid system, that there's other receptors that are completely different. The TRPV1 is an example. They're called the vanilloid receptors that have completely different functions in your body, but CBD seems to activate them. And then I wanted to also talk about clinical research that we have. So this is a great review. It's basically a meta-analysis that provides evidence of benefits and information about the potential for adverse effects, so AEs. And the authors here, they reviewed over like 23,000 abstracts, over 500 full text articles to compile this data from randomized clinical trials, right? So these were humans in clinical trials, and they compared cannabinoids to placebos for many conditions. And so the reason why I'm calling this title Cannabis and Humans on this slide is because they weren't specifically just looking at phytocannabinoids. And those absorption slides we just talked about were looking at individual phytocannabinoids, where here, some of the interventions, so some of what was being taken was smoked cannabis flower. So we can't dismiss that some of these effects were from things like terpenes or other chemicals in the plant. And then some of the studies, they were using this synthetic THC nabalone and then the sublingual THC CBD mix, and they were comparing them to the placebo. But basically the summary of the results is they found improvement in pain when compared to placebo when using cannabinoids, and that cannabinoids were associated with approximately a 3-time increase in odds of any adverse effect when compared to the placebo. And the higher the number that they give is like the more likely that you would have a negative impact. And some of the examples of adverse effects were dizziness and dry mouth, but they were not fatal. And the next 2 slides, I basically didn't want us to forget as we discussed phytocannabinoids and also affect us. The secondary metabolites in cannabis, like terpenes, have been reported to affect the human body. For example, myrcene, which is also in hops, is reported to have a pain relieving effect. And then our next slide is going to tell us that alpha pinene, so if we advance that slide, there's another example, has a potential antibiotic effect. Catherine, I think you advanced and then it jumped back. There we go. Thanks. And so I'm not going to go through these studies and talk about the data that we see in them. It's just, there is literature out there, you know, some of it is older, some of it is current, and it's looking at how the terpenes do affect the human body. And then finally, I do want to mention that we have synthetic cannabinoids legally available in the US, right? We have dronabinol and its analog nabilone, and there are two examples of synthetic form of THC. And then we have the recently approved Epidiolex, approved in 2018 for certain conditions, which is derived from naturally occurring CBD. And I imagine you've had lots of policy talks, and this is basically the reason that you know, you can't market and list CBD because it is considered an investigational new drug and an approved FDA drug. So in order to, you know, market and sell CBD, you would need to go through the normal channels of new drug development. So hopefully this is what you were hoping for to learn today. If you advance the slide, that would be great. How different cannabinoids affect different symptoms and ailments. And we also have a cannabis plant biology program that you can earn certificates for at UVM. So if you're interested at all, if you advance the slide one more time, there's these websites that you can see. So Catherine, just advance it one more time. This is the website. And then finally, I know we only have a few more minutes, but if anyone has questions, I'm happy to answer them now, or you can always email me as well.
Awesome. Thanks, Monique. That was great.